It's now our pleasure to welcome today's keynote speaker, a scholar whose work moves effortlessly across the boundaries between biology, philosophy, and the public understanding of science. Professor Telmo Pievani holds the chair for the philosophy of biological sciences at the University of Padua, the first of its kind in Italy, and has long been one of Europe's most prominent voices reflecting on evolution, human diversity, and intellectual consequences of modern biology. He has published more than 400 scientific and public works, and his forthcoming book is titled All Possible Worlds. And there is one more delightful detail. In 2024, the International Astronomical Union named an asteroid after him. So tonight, if the skies remain clear, you might look up and remember that somewhere out there, orbiting quietly through space, is asteroid Telmo Pievani. But now we're fortunate to have the original here with us, talking about the challenge of de-extinction, a bioethical critique. Welcome, Professor Telmo Pievani.
Thank you. Thanks so much, Ralph. And I have to underline that the asteroid with my name is not one of the asteroids with the risk of impact on the Earth. So don't worry. I would be surprised to give the name with an asteroid killer of the humanity with my name. Would be a shame. So thank you so much for this invitation. It's an honor and a privilege to be here for sharing with you this keynote and this morning. And thank you, Ralph. Thank you, Stephanie Dion, for all the help for the conference, and Monsignor Renzo Pegoraro. It's always a pleasure to be together discussing about these topics. In Padua, at the Department of Biology, I teach also ethics for biotechnologies. And we decided, the Department of Biology in Padua, and we decided to propose to our students the new course of ethics for biotechnology as mandatory for every student in the third year of the first Bachelor of Biotechnology, because we decided and we thought that today something is running too fast, even for our students, that in few years after the master degree, they have to use new devices and completely new tools for managing and for manipulating life. So this morning, I would like to share with you a bioethical discussion on a particular case of engineering life that could seem quite crazy and very specific, but I think it's very interesting in order to introduce some topics, some general topics. It's the so-called de-extinction project, brought technically closer in the last 2-3 years thanks to the rapid developments exactly of gene editing, so the copy and paste of the DNA. This is a limited but very emblematic case because it illustrates very clearly the implication of the biotechnological acceleration we are living today. It had strong media impact the past year, 2025, and this serves careful analysis on ethical, epistemological, and social levels. So it's very specific, but it refers to general theme, a philosophy of new technology, of the new biotechnology that I would like to challenge this morning, that is emerging, especially in the United States now. The word de-extinction seems very positive in itself, because it's the opposite of extinction. It means bringing back to life individuals belonging to an extinct species, often extinct because of us, driven to extinction by our ancestors. It seems like a struggle against an unescapable fate, like an atonement for a sin we are committed in the past. But if we look closer at what is happening in some private US labs today, and maybe elsewhere, the situation is much more complex. So please, Let's ask ourselves, what kind of research is this? In April last year, a private Californian company, Colossal Biosciences, announced the de-extinction of the Aenocyon, the wonderful dire wolf made famous by a very successful saga in TV. The Aenocyon is actually a magnificent carnivore that disappeared from Earth more than 12 millennia ago. And of course, the news received great media coverage, but no one was interested in the original Aenocyon, an otherwise charismatic animal from the Pleistocene, the dire wolf of North America and East Asia, with a deadly bite that we can compare with the bite of saber-toothed tiger and many other great predators. Its genome was really extracted from fossils and sequenced completely in 2020, so 6 years ago. The news made headlines around the world because an extinct species, we said, and we heard about, had been resurrected. Let's note one important point, very important for me as a philosopher of science. This research has not been published in a scientific journal, has not been discussed in any scientific conference, and has not been approved by any ethics committee of any research institution. Nothing. It's been just announced on social media and then in the general media. This is something new for me in the scientific community. Anyway, the scientific content of this news, however, is completely wrong or completely marginal. Why? Because no species has been de-extinct or resurrected. Nothing. Technically, it happened that genetically modified dog puppies were born, dog puppies, with certain traits of their DNA modified to make them somewhat resemble to the Pleistocene Aenocyon, through the insertion of 6 or 7 Aenocyon genes through gene editing. And indeed, a month later, in May 2025, the press office at Colossal admitted that no dire wolf had been born, but rather grey wolf pups, so the ancestors of all dogs, with a handful of genetic modifications. But meanwhile, donations in a month exploded. A very clever technique, but absolutely unfair. Of course, the strategy worked very well, given that the company is gathering now millions and millions of dollars of donations. So, for now, Aenocyons remains a fossil, with its DNA well preserved, its evolutionary history, and its extinction in North America, which coincides exactly with the arrival of the first Siberian hunter-gatherers across the now submerged continent of Beringia. So a wonderful story. But now we can put the points together. We connect the points. It's safe to bet that this line of research won't stop. Indeed, a month before the dire wolves, maybe you remember this photo, in March 2025, Colossal, the same company, announced the birth of some mice with mammoth fur, modified on 9 genes that actually directed hair growth in mammoths. And so we understand here the big target, which they hope to reach, according to the press office, around 2028, so in 2 years. The mammoth, the beloved and charismatic mammoth, which also became extinct at the end of the Pleistocene, except for a small draft population that survived longer on Wrangel Island in eastern Siberia. The park to house these creatures is ready in Siberia, in the Kolyma Valley, and they have called it, with little imagination, Pleistocene Park. So everything is ready to do that. The technique, anyway, is certainly more promising than the reverse breeding. You see here the traditional attempt to recreate extinct species. For example, to revive the original European bovine, the aurochs, which became extinct in the 17th century. Its distinctive genes were identified, of course, and attempts were made to select them in existing cattle breeds, essentially trying to turn back the clock through artificial selection and, in some cases, hybridizations. But it doesn't work. And so far, this hasn't been successful. The genome, not only human genome, animal genome, is a recalcitrant, very complex network of connections. So nothing in this line. The new technique about mammoths is also certainly more promising than cloning, you remember, which has been attempted in recent years to de-extinct the Pyrenean ibex and the American passenger pigeon without success. In those cases, all the DNA of the extinct species must be extracted from the fossils and inserted into the egg cell of the living donor and nurse species. But it doesn't work, likely due to the state of conservation of the extinct species DNA and maybe due to the same technique of cloning that is very disturbing for the DNA. In other cases, such as the Australian thylacine, which became extinct in the 1930s, or the New Zealand moa, which disappeared half a millennium ago, a sufficiently close cuisine species is lacking. So in this case, it's not possible, no way. But now we have the gene editing, so versatile, precise, and cheap. And so we have the mammoths. This is the technique. So we have to understand the technique very precisely, and then we can discuss ethically. The de-extinction project, literally reversing the extinction process, involves taking the genome of the mammoth's closest living cousin, the Indian elephant, and matching it letter by letter, nucleotide for nucleotide, to the mammoth genome. So it's a completely new technique. We know the mammoth DNA very well, of course, having extracted it from specimens very well preserved in the permafrost, including some soft tissues, and fully sequenced it. So we know where the differences lie between the mammoth DNA and the elephant DNA. At this point, thanks to gene editing, we can make elephant DNA increasingly similar to mammoth DNA, until the time will come, we don't know when, to fertilize a female elephant with this mammoth-enhanced DNA. The result will be an animal with mammoth phenotypic traits, even though its mitochondrial DNA will be that of its mother, of course, and the influences during the pregnancy will also be elephant-like. An alternative, more difficult, may be to develop the embryo in an artificial womb. In short, a mammoth-elephant hybrid will be born, something like a mammophant. This is why full woolly mice and dire wolves are intermediate steps towards this end, the mammoths. Colossal, what is doing Colossal? In Colossal, they want to understand the effect of inserting genes from other species and producing modified offspring. So far, it's working. So it's technically possible, even though, admittedly, nothing has been resurrected so far. Furthermore, we don't know how the mammoth's genes interact with each other to produce the mammoth phenotype. And there's no guarantee that modifying some genes in the Indian elephant will produce a mammoth. We are still too ignorant about the complex regulation of genomes. And these are the guys involved in this venture. Behind this research, there clearly lies the post-humanistic philosophy currently dominant in the Silicon Valley, an unlimited and arrogant faith in new technologies to modify human and other animal DNA, to shape ecosystems, to enhance humanity for a privileged few, and our brains. Another research everyone is talking about today, maybe we will discuss it this afternoon, but has not been published yet in any scientific journal, is Neuralink. The new trillionaire techno-feudalists display great confidence in the miracles of technology and have decided to conduct this research privately, outside and against all the rules the scientific community has built over the last 2 centuries. Our rules are imperfect, we know, but very precious. Transparency, data sharing, peer review, analysis of social, ethical, and economic implications of new discoveries and devices. All of this here has been replaced by marketing, by unregulated competition between companies, and by charismatic figures who monopolize AI and social media algorithms. So the de-extinction of an elephant-mammoth hybrid, in any case, is now technically possible. And perhaps we will hear about it very soon. But what motivations do these researchers give for undertaking such a venture? There are two motivations. The first one is to mitigate the current biodiversity crisis by reviving extinct species. The second one is to use these resurrected species as ecosystem engineers, supposedly restoring Pleistocene habitats and ecosystems. These are the two official motivations for this research. Well, for those who study evolution and bioethics applied to ecology, these two arguments are completely inconsistent. Now I'm trying to explain why. Not only there's no resurrection here, but since those cubs are merely genetically modified hybrids, but also there's no original environment into which we could release them, because the Pleistocene ended 12 millennia ago and will never return. Colossal claims that revived mammoth herds would reduce permafrost melting and restore the Pleistocene tundra ecosystem. But the hypothesis is completely untenable. In the cold environment where Aenocyons once lived, temperature now reach 28 degrees Celsius for 6 months of the year. 28 degrees. Pleistocene is gone forever. There's no possibility that these Aenocyon hybrids or the future mammoth hybrids they are planning could restore past ecosystems. This justification is devoid of any scientific validity. Furthermore, these poor creatures in the future, which will become freaks for Pleistocene amusement parks for rich people, will encounter an environment full of pathogens that didn't exist in their time. Ignoring all these caveats, this is the news, very new, Colossal has now promised to bring back to life the mythical dodo, the flightless bird, so that its ecosystem can be restored in Mauritius. Okay, so regarding biodiversity, we can check. This is the latest paper published 2 months ago, in January, by one of the world's leading experts in biodiversity, Tony Barnowski, Berkeley. The thesis here in this paper is dramatic. Holocene biodiversity is gone forever. This is the biodiversity of the Anthropocene. So some data. The vertebrate extinction rate in the last century has reached 100 times the normal rate in evolution. There has been a 73% decline in wild species population in the last 50 years. The data on insects, including in pollinators, are even worse, even in Europe, especially in Europe. And the paper speaks about an insect apocalypse. A biotic homogenization is underway, caused by land use, intensive farming, and translocated invasive species. The anthropogenic mass, that is the weight of all human artifacts, exceeded the global biomass in 2020, 6 years ago. It means that there are now more human objects on Earth than animals and plants. More human objects than animals and plants. Parks, reserves, and protected areas are, according to Barnowski, Holocene relicts. They cover only 70% of the land, 80% of the ocean, and the connectivity between them is less than 10%. Given this evidence, do we really think that the presumed resurrection of 2 or 3 species can reverse the biodiversity crisis? And can we seriously think that this species can recreate Pleistocene environments? It makes no sense. And Barnowski agrees with that. This is also what Barnowski thinks. If you see here, this is interesting because it's exactly citing De-Extinction and he calls it a false promise. And as you read here, species are products of evolutionary and ecological processes that result in functioning ecosystems made up of organisms intimately connected through webs of interactions. And if you read here, gone forever are the learned behavior, diets, predator or prey, and microbiomes that accumulated over millions of years. So according to Barnowski, and I strongly agree with him, we need an ethics of Anthropocene, a new ethics for Anthropocene, including humanity as a part of the story, not only has the species that produced this destruction of the biodiversity, but now can be the shepherd of the steward of the change. According to the IUCN, the International Union for Conservation of Nature, the point is very clear. De-extinction is not a sensible strategy to mitigate biodiversity extinction. You see in this document, this was the first document related to de-extinction. Rather, according to IUCN, it's a way to distract attention from what we really need to do. Stop deforestation, the spread of invasive species, land consumption, pollution, and indiscriminate hunting and fishing. Not even tree ferns alone could recreate a past ecosystem, because habitats are highly complex network systems. Evolution cannot be regressed. We move forward. With the money being wasted on de-extinction, we could save dozens of species that are not yet extinct, but are on the brink of extinction due to human greed and the devastation of tropical and equatorial ecosystems. Furthermore, there's also another ethical point. You can see here a clear taxonomic bias, because we only want to revive charismatic and easily selling species, usually mammals like ourselves. But the real, very important ecosystem engineers are not always animals like mammals. They are insects, microbes, and so on. We don't have a sufficient understanding of the genome to control for all the unintended ecological effects and epigenetic effects. Finally, the technique raises serious animal welfare concerns. These proxies of extinct species will be novel species in the environment. They could hybridize with existing species. They could become invasive species. They could suffer from recently evolved pathogens that their immune systems are unfamiliar with. We could reach, according to IUCN, the paradoxical situation of de-extinguishing a species only to re-extinguish it immediately afterwards. So, final analysis, it seems very clear. It won't be a mammoth, but a mammoth, so a functional mammoth. So we have a problem, a great ethical problem of science communication. So we have a problem of communication ethics. The target number one, counteract the 6 max extinction, makes no sense. Rather, with the same biotechnology and money, we could save really endangered species, for example, by increasing genetic variability and defend the rights of native peoples who live with them. The target number 2, counteract climate change through reshaping the Arctic ecosystem and slowing down permafrost melting, makes no sense. It would take too many animals for too long for too much money. So the costs and risk far outweighed the presumed benefits in this research. What unexpected mutation can we imagine in this mammuthification of an Asian elephant? We have an ethical problem of unintended ecological effects. And finally, what new pathogens will these animals encounter after 10 millennia? So we have an ethical problem regarding the welfare of these resurrected animals. Well, despite all these bioethical problems, there's currently no international regulation about this research. Even if an international framework of bioethics and biolaw existed, the United Nations would not have the power to enforce it right now. International law and multilateral negotiations are at their lowest point since the Second World War. And meanwhile, biotechnologies like AI and many others are running faster and faster. The scientific community has its own self-regulation rules that can work, through journals, conferences, and public research institutions and ethical committees and so on. But this research is often conducted by the military sector and by unregulated, business-oriented private entities without any rules, so outside the scientific community. And also, as an evolutionist, I have to add that manipulating the germline by modifying animal embryos means introducing heritable and therefore evolutionarily significant and irreversible variations into a population. Put in another way, with CRISPR, Homo sapiens has invented a new mutagenesis tool, so by technological means we become a direct evolutionary change factor for ourselves and any other biological species. We are an evolutionary force. This has never happened before in the history of human technologies. In March 2018, Sheila Jarzanov, that has been hosting a previous conference by the Pontifical Academy, and Benjamin Harbat proposed in Nature the establishment of a global observatory on genome editing, modeled on other international networks such as the one on global warming, the IPCC, in order to promote a wide-ranging dialogue between science and society on these biotechnologies, receiving, unfortunately, few reactions so far. Nothing. According to the scientific community, anyway, the bioethical assessment of de-extinction is completely negative. Before concluding, however, I would like to add two examples of gene editing application that make the picture even more complex. The first may seem crazy, but it's a real research project in the United States now, in Montana, recreating a dinosaur-bird hybrid. They call it Dino-chicken. The guy here, he's Jack Horner, 79 years old now, and has been collecting dinosaur fossils since he was 8. He's great. In Montana, he is something like an Indiana Jones of the paleontology. He filled the Bosman Museum there with wonderful T-Rex and many other fossils. He's one of the leading experts on the embryonic and postnatal development of dinosaurs. And 3 species will forever bear his name. He was also the scientific advisor of Jurassic Park, the movie. Do you remember? He wrote the text. And remember the raptor expert paleontologist who visited the dinosaur island? First ecstatic, so happy, then perplexed, and finally terrified in Michael Crichton novel and in Steven Spielberg's first film in 1993. That's him, Jack Horner. Okay, over the past 30 years, that's the serendipity of this line of research. Paleontologists like Horner have gathered many discoveries that profoundly changed our understanding of dinosaurs' phenotype and behavior. The most surprising development is that dinosaurs, as we know now very well, were much more similar to birds than we had previously assumed. They were vividly colored, and namely theropods had feathers. They are ancestors of birds. So living birds are living dinosaurs. That's the idea. Hundreds of characteristics unite the two groups, and in fact, with the only exception of flight, all the traits we thought had evolved in birds were actually already there in dinosaurs. With different functions, maybe, because evolution likes reuse and tinkering, but they were already there. Hollow bones, feathers, the fusion of clavicus into a furcula, the hex, pneumatic chambers in the bones, parental care, and so on. All of this evolved in theropod dinosaurs. This is where the crazy idea was born. Horner and a colleague at the Montana State University, geneticist Dana Rashid, are working on a project to resurrect some trait of dinosaurs to the genetic modification of chickens using gene editing. However, we don't know the genomes of the avian dinosaurs like in Jurassic Park. It's not like with mammoths, because ancient DNA is not preserved for such a long time in bones, for tens of millions of years. The technique is a little different. So they tried to find in birds' genome today the atavistic genes that are ancestral genes that has been switched off during evolution, But that can be reactivated, and we know this because, for example, in some snakes, sometimes these genes are reactivated and the snakes are born with legs, for example. So they began to look at the atavistic genes in chickens, and they were able to do them, to find them. In 2009, Matthew Harris, University of Wisconsin, discovered an atavistic gene for teeth and produced the first bird with tooth fragments in the beak. A lab in Chile succeeded in resurrecting the fibula bone in a chicken leg, and another lab in Yale University altered a chicken skull to the point where it more closely resembled a dinosaur one. And Horner himself, Tim, in Montana, is working on the genes of the tail of the dinosaur in order to recreate the tail of a dinosaur in the chicken. At this point, you might think that this is a waste of money for a highly controversial purpose, exactly like the de-extinction, and I agree with you. But here is where true serendipity comes in this story, because Dana Rashid, who follows the progress from the genetic side, completely by chance, studying the genes of the definition of the vertebrae in birds and dinosaurs, identified the probable genetic cause of a highly debilitating human spinal disease that we call ankylosing spondylitis. Further research is needed to confirm this hypothesis and to determine whether there's a way to genetically cure the disease. But the very fact that this potentially significant evidence for human health emerged while attempting to recreate the Dino-chicken is thought-provoking for me. So before condemning and banning research, we should always remember how ignorant we are and what we can lose precluding ourselves from the possibility of important discoveries. Anyway, now Horner is looking for a donor to breed the Dino-chickens on an island. And let me say, let's hope the story doesn't end like in the movie. So we have to check in the future. My final example is, I think, the most impressive, is the de-extinction not of a species, but of a single genetic mutation. And this refers to humans. So it's quite different, not other animals. Truly impressive. A team of researchers at the Max Planck Institute in Leipzig, led by Nobel Prize winner Svante Pääbo, a great molecular anthropologist, attempted to verify the effect of a different genetic mutation between Homo sapiens and Neanderthal. So here the story is very serious, is related to our research in order to understand the genetic differences between our species and Neanderthal species. But they did it in a completely revolutionary way. Why? This is the paper in Science with the description. Compared to sapiens, Neanderthal, we know that already, had a single different mutation in a very important gene, TKTL1 gene, which is linked to brain development. The mutation is very tiny, it's just the substitution of a single amino acid in the protein. So you see there, a lysine and an arginine in the case, an arginine in Homo sapiens, a lysine in Neanderthals. So what the researchers did in this case, the researchers created a human cerebral organoid in the lab, human organoid from Homo sapiens cells, a sketch of a brain tissue, which is now normally used in medicine. Then, through gene editing, they engineered the organoid by inserting the Neanderthal mutation in the TKTL1 gene. And they were able to observe that the density of the dendrites indeed decreased significantly in the organoid. So it's a wonderful discovery, so with a cover in Science, so it's great because essentially they verified in the lab the actual phenotypic effects of a mutation which occurred tens of millennia ago in another species and then occurred in a different way in Homo sapiens. So now, for the first time, this never happened before, we can, in the lab, recreate a mutation of the past in a tissue, in a modern tissue. So they resurrected a mutation. But in doing so, they also created a Neanderthal brain organoid in the lab. And this objectively worries us a little bit. So for the first time, we have in the lab a tissue of Neanderthal recreated through gene editing. And someone else could think that they can go further. Other mutation of Neanderthals in other tissues, and now it's technically possible to do that in the lab. So concluding, summing up, so addressing these life engineering challenges will require great wisdom for me, humility, and responsibility for the good of humanity. These are the 5 ethical principles that the Veronese Foundation Ethics Committee, Luigi Ripamonti knows very well our research, is a colleague of mine in this committee, in order to address these cutting-edge cases. We deal with cases at the frontier of biotechnologies. So non-maleficence, beneficence, justice, freedom of research, responsibility in research. And I think, just as a conclusion, that rather than towards the creation of new humanity, like post-humanists think, gene editing should be directed only towards improving the conditions of this suffering humanity here, guaranteeing fair and universal access to the benefits offered by these new biotechnologies. Ultimately, it would be another challenge for our democracies. Before the mammoths return among us, let's take some time to understand where these private biotechnologies are going. To refine, integrate, and adapt these principles, I believe dialogue between different cultures, between different philosophical and religious sensibilities will be crucial, giving voice, mostly for me, to those who have not had a voice until now, in a mutually enriching exchange. So therefore, I'm once again, Ralph, grateful for this invitation, and I'm sure I will certainly learn a lot from the works of these 2 days. So thank you so much.
Thank you very much, Professor Pievani, for this fascinating talk, giving us insights into the challenges of de-extinction. We have time for questions here in the room and via the chat. My colleague Alexander Benyak is in the back. He has a microphone, so I'm sure there are plenty of comments or questions. Alexander, maybe in the back, the lady there. Yeah, okay.
Yes, thank you so much. It was indeed fascinating, really. It reminded us what's going on and also opened the window, the wide window to the new projects that are developing. I wanted to ask you something that is really a concern of mine for a long, long time. For these last decades, we have been trying to find a balance between scientific development, technological innovation, and ethics as the voice of the citizens in a democratic societies. So this is what some of us here have been committed to. But nowadays we are facing a new world characterized by deregulation, by the voice of the markets, as you very well pointed out. When you talked about Colossal, I immediately thought also about Neuralink that was going to bankruptcy when they started to announce the Neuro device. And all of a sudden, they gained so much money on stock. So, okay, it's market. So we can agree on these 5 or other major ethical principles. We can have them as a consensus platform in small communities. But what can we do at the international level when deregulation is the most important orientation nowadays? Even the European Commission in, for instance, the life sciences strategies, they are heading smoothly, talking about simplification.
Okay, the question, please.
The question is what we can do. I'm sorry, I was really excited about this. Thank you. What can we do when we are talking about
the answer is
regulation?
I don't know, of course, but thank you, Alessandra, because I totally agree with you. I think that what we have to discuss, that's my proposal for today and tomorrow, is the fact that for people like me working in a department of biology, working with biotechnologists, they know that the landscape in 3 years, in 2 years, completely changed for our research. As biologists, as biotechnologists. Why? Because we are in a situation in which international rights are completely underlined, so it's completely moving away. We have an acceleration of the power of these technologies. We have new actors in the landscape, private actors. These private actors, without any regulation, they have a lot of money, but they have also great media success. So it's a completely new situation. So I have just an answer for you. I think we need new alliances. We have to be together in the public universities, international public institutions, religious sensibilities. We have to environmentalist movements. So we have to find new alliances. As Pope Francis wrote many times, we need new alliances because the landscape is completely changed. So we have to think with new categories because other guys now started to think with new categories. So because when you find people announcing in social media that they recreated the mice with the fur of a mammoth, the first question in all my life have been, in which journal did you publish this research? In which conference did you discuss this? Which ethical committee? So let me know. And so now we are completely overcoming these rules of the scientific community. We have to be very serious about that.
Thank you very much. This is really fascinating discussion to have. Just for those on Zoom, this is Kevin Fitzgerald from Creighton University. There's been a desire in the genetic community for decades now to have this global dialogue, right? There's been several events that have occurred. Dr. He, back in 2020, whenever that was, when he announced genetically engineering human embryos. But it never happens. So the question I guess we face is why doesn't it ever happen? What are the logistical issues? But also more fundamentally, when you request this dialogue, are you looking for people who all share similar understandings and appreciation for the principles that you listed? Or are we going to include those people who don't agree with that and want to move ahead as fast as possible? And you're going to run into issues where, for instance, the technology does in part approve or provide what you asked for, benefiting human health, but does so by genetically engineering pigs so that they can provide human organs, right? So we're, you know, there's several things here, but I'd just love to get your thoughts on that. Thank you.
Yeah, I agree. Let me answer just in two ways. One is, every time I meet more than 200 students in my lessons, and this makes me a little bit optimistic because I think that in our new generations, something is changing in terms of the sensibility toward the public aspect of the research. When I discuss with them about de-extinction, and at the end we vote, and the vote is 98 against, so 98% against any possibility to approve any research like this from biotechnologists people, and 2% are positive. Never happened before. Before, in biotechnology, there was a much more liberal attitude to this technology. Now something is changing in the sensibility of young people. 2 years ago in my department, I decided to provoke my colleagues with a vote, a secret vote about the possibility to go further in the direction of the enhancement of viruses. Maybe you know this other line of biotechnological research. So enhance virus in lab in order to prevent mutation that viruses can have in the environment. And more than 70% of my colleagues working on biotechnology were contrary to this application, contrary a priori, so since the beginning. So we have not to do that, because in this case we will have in the lab weapons, biological weapons. And that's my final point, that it's something like a paradox. Now you can find in great journals, it happened even last year, editors of the journals saying, okay, in this issue, we will publish this paper about biotechnologies. We know that it's very controversial in ethical terms. We know that in some nations this research is illegal, but we decided to publish because it's better to know than not to know. And you see here a paradox between transparency of science and ethical issues. Never happened before the pandemic, for example. Never happened that editors publish an issue saying, okay, I know that this is illegal in many nations, but I decided to publish it because it's better to know. That's typical of a situation of dramatic contradiction within the scientific community. I think in order to answer again, what can we do? I think that we have to stress much more the positive and humanistic application of these biotechnologies and be very radical against this post-humanistic, millenaristic approach to technologies that you can see even in the case of AI. Maybe you have seen in PNS a few months ago this paper by 2 microbiologists saying that in the future AI will become a symbiont in humans, and we will have a new step in human evolution with this symbiosis between humans and AI. And according to the symbiosis, we will have a division of labor. And if you see it in this paper, in the final chapter, the division of labor in this symbiosis will be that humans, we will keep the physical function, reproduction, eating and so on, and AI, we will have the motivation, decisions, memory, so what is interesting in life. So, and then in the end they say, it could seem a little bit dystopian. Yes, it's dystopian. But now you can find this paper in PNS, seriously debated in PNS. So this is typical of this philosophy, for me, that is emerging and is spreading in some part of the scientific community, post-humanistic, so this techno-millenarism, according to which we will have a new singularity in evolution. I never believed in singularity. We will keep humans in the next steps of evolution, and I hope so.
Honoring our program, we have time for one more question. Laura, maybe?
There is also a question from the chat.
Okay.
Sorry, I will be very brief. Okay.
Yeah,
Make
very
it
brief.
quick. Mm-hmm.
So basically, it's the question. I paraphrase it because it's very long. But could the very framing of our era as one requiring active human management of nature be part of the problem, encouraging interventions such as species resurrection, rather than promoting restraint or trust in long-term ecological self-regulation? So we are telling we need to do something. So when people come up with some ideas, we are then kind of against it. So I guess
that's a very important question. If you have seen the paper by Barnowski in the Proceeding of the Royal Society, that's exactly the point, and it's very interesting. Because now in the ecological attitude, we are seeing something like a paradigmatic shift. I think it's very interesting. So from the idea that in order to preserve environment, we have just to conserve and protect. That is still, of course, true. But maybe Barnowski and other guys are starting to think that it's not enough. We have to conserve, preserve, but we need active intervention of humans in order to restore, recreate environments. But Barnowski, of course, wrote, okay, we have to actively be steward of the biosphere, but with an ethics of the Anthropocene. No, it doesn't imply de-extinction. It implies that you have to recreate the condition in order to make biodiversity able to restore. So we agree about the fact that we need an active intervention of humans that is still different to the traditional and classic environmentalist thinking, okay? But de-extinction is another topic.
Last question, Professor Palazzani, please.
Thank you really very much. I was thinking about, of course, regulation. We need, but regulation, it takes a lot of time to arrive, and it takes a long process, and the technological evolution is very rapid. What do you think about education? I mean, raising awareness on scientists, because the real problem is, you talked about companies, of course, they are the main stakeholder, but also scientists, because without them, companies cannot do anything. I'm thinking about moratoria on gene editing on germline, maybe something similar that comes from raising awareness, ethical awareness in scientists. What do you think about?
Sure. About the moratorium, maybe you know, maybe we will discuss in these 2 days. They could be a solution, but it's in the international situation that we see today. When you propose a moratorium, you have to have the tools to apply it and to make it respected. And in this situation, it's very, very hard to think that nations that are completely, including the United States, they are moving away from all the international organization for the first time in the history. So it's something completely new. So in this situation, you can approve in the United Nations a moratorium, but the United States themselves will be the first to not respect to the moratorium. So we are in a completely different situation. So I agree with you. We have to work in education, but education is slow, and we have to wait for new generations. And I'm optimistic because I see a new generation of scientists, like the students that I have, that, for example, they want to work in public institutions. I think it's very important, this idea of public, to work in public universities, to work in public institutions. I don't want to say that private companies are wrong or are always bad, but I think that we need public today. We need to work in public situation with transparency and balances and checks in this research. And then we have to, of course, to write new rules, but we have to be very rapid, because in few years we will see a lot of surprises coming from this line of research from private companies.
Thank you very much.
Sorry, I'm here anyway, so because I have two other questions, so
we
in the coffee break.
We have to take a break here and be back on time at 11 with our first panel on science and human beings. Thank you all for your questions, and Professor Pievani, thank you.